What if I told you that this blue dye, yeah, the one used in aquarium tanks, might hold the key to fighting Alzheimer's and Parkinson's disease? Methylene blue has been around for over a century, and while I have used it countless of times in the lab as a dye, neuroscience is now buzzing about its potential to treat some of our toughest brain diseases. So how does a simple dye become a candidate for therapy? Well, give me 10 minutes of your time to put my neuroscience PhD to good use. And I will distill the scientific literature of what is methylene blue, how it works in the brain, and the real controversies around bringing it to the clinics. So, let's dive in. Hi there, I'm Yvette, a neuroscientist and an author of several neuroscience studies. On this channel, I like to break down how the brain works, decode its mysteries, and explore the deeper questions that they raise about who we are and how science will shape our future. So, if this sounds like something right up your alley, you know what to do. So, let's rewind a bit. Discovered in 1876, methylene blue initially gained popularity as a stain for microscopy, vividly creating contrasts of structures in tissues and cells. You probably already have used it in your high school biology class to stain some cells under a simple light microscope. Well, if Giemsa stain sounds familiar, yeah, you did. Anyway, early pioneers like Paul Ehrlich and also Ramon Santiago y Cajal used it as a supravital stain for nervous tissue, a practice which continued until decades later. Interestingly, Nobel laureate Paul Ehrlich also used it to treat malaria, and it soon found medical application for other blood diseases, urinary tract infections, and also served as an antidote for cyanide and carbon monoxide poisoning. Interestingly, an early study in 1961 by Lindahl and Oberg suggested that methylene blue could inhibit fish death caused by pesticide leakage in the water. While it is now one of the reasons why aquarium water is blue, it also hinted at methylene's therapeutic metabolic potential. In fact, it was used clinically as a neuroprotective agent against drug-induced encephalopathy. And recently, neuroscientists have rediscovered this dye due to its significant interactions with the nervous system and cellular metabolism. But actually, how legit is all of this? Imagine your brain cells as part of a metropolitan city. Like New York, for example. There's loads of things going on, loads of action. And for all this action to function properly, one needs energy. Loads of it. And similarly with brain cells. In the cells, this energy is generated by tiny powerhouses called mitochondria. And studies have now shown that these mitochondria become impaired in diseases like Alzheimer's or Parkinson's disease. And due to their tiny but mighty property, this impairment leads to cell starvation and widespread damage. And here methylene blue plays multiple critical roles, specifically in these four areas. So the first one, methylene blue is highly effective at crossing the blood-brain barrier, which is a notorious barrier to cross as it is highly selective. In fact, some of the drugs do not make it to the market, not because of their ineffectiveness, however, it is due to their inability to cross the blood-brain barrier efficiently. And in the case of methylene blue, well, its early use was as a nervous system dye. So as you can imagine, it can readily cross the blood-brain barrier and it concentrates in the nervous system tissue, specifically into mitochondria. And this is essential for its therapeutic effects. Secondly, it supercharges mitochondria by restoring their energy production. So if we had to go down to the nitty-gritty details, methylene blue acts as an alternative electron carrier, bypassing mitochondrial complex blockages, reducing harmful electron leakage, and as we've mentioned earlier, restoring energy production. Lol, I know, that was a handful. And if this detail was too much, just let me know in the comments down below and I'll refine for future reference. Then the third point is that it acts as an antioxidant, as it is able to reduce harmful molecules known as free radicals, which cause severe damage in our brain, and this action can prevent neurons from dying. And the fourth point is that it prevents these toxic protein clumps, which are hallmarks in neurodegenerative diseases, like for example in Alzheimer's disease there is tau and amyloid beta deposition. So as you can see from these 4 points, methylene blue is a great therapeutic candidate for Alzheimer's disease, because in short, it decreases the bad stuff and increases the good stuff. But these therapeutic properties are not limited to Alzheimer's disease. For example, in Parkinson's disease models, it enhanced mitochondrial activity, improving neuronal survival and function. Beyond these diseases, it has been shown to have neuroprotective properties in traumatic brain injury, ischemic stroke, optic neuropathy, and Huntington's disease, making it a broad-spectrum neurotherapeutic candidate. So, methylene blue sounds impressive, right? And yes. However, if it is such a remarkable compound, why isn't it mainstream treatment yet? Well, and the answer is that there is more to the story than meets the eye. As exciting as methylene blue's potential seems, clinical trials have produced mixed results. It was described to have a hormetic dose response, which means that low doses are beneficial, intermediate ones are ineffective, and high doses potentially harmful, causing conditions like methemoglobinemia, ironically a condition that it supposedly treats. And this is all because methylene blue's mode of action is quite complex. At certain concentrations, it paradoxically increases the reactive oxygen species produced, contradicting its usual antioxidant role. It also presents distinct effects on mitochondrial respiration, depending on dosage and cellular context. And all of this suggests unexplored pathways of action. For example, in Alzheimer's disease, while methylene blue was shown to inhibit tau aggregation, recent findings show that it promotes tau phase separation and gelation, a mechanism which is quite distinct from simple aggregation inhibition, and thus complicating therapeutic predictions. Clinical translation remains challenging, partly due to the intricate nature of neurodegenerative diseases, and partly due to the discrepancies between promising preclinical findings and mixed human trial results. For example, in clinical trials with methylene's blue derivative LMTM have yielded mixed outcomes, highlighting variability in effectiveness among patient groups. Yet, researchers remain optimistic, continually refining dosages, formulations, and exploring new applications like photodynamic therapy, which combines methylene blue administration with targeted light exposure. And this is the mode which seems to be provided by a selected few clinics. However, this complexity leaves us wondering, is methylene blue truly a groundbreaking discovery? Or is it just another example of hope outpacing reality? So, if you're anything like me, you're pretty intrigued by this chemical. And you're wondering, why do we not hear about this more often? I have to say that I came across the use of methylene blue in neurodegenerative diseases, not through my work as a research neuroscientist, even though it's my business, but by browsing through YouTube. Weird, right? Anyway, I was watching a video from one of my favorite creators called Chase Hughes, and he mentioned the use of methylene blue in brain diseases. Now, I usually don't like to listen to randos actually talking about potential new treatment in the field of neurodegeneration, because, well, there are many people out there who are really desperate to find a cure for themselves or for their loved ones. And unfortunately, there are many predators out there who are ready to prey on this and sell scams. However, in my opinion, Chase didn't seem to have anything to gain from spreading false information, as he's not selling the treatment himself, and actually had a very genuine reason to share this because it is his personal experience and he used it himself to battle his own brain disease. So if you want to take a look for yourself, I'll put a link to his video in the description down below, and you can check it out for yourself. And so his video actually prompted me to dig deeper into methylene blue. And what I shared with you today is the distilled version of the scientific literature that I came across. Like with many treatments for brain diseases, it is quite tricky. And in my opinion, this is primarily because we still do not understand fully how the brain works. And so, interfering with a system, a really complex one, and one doesn't understand fully how it works, well, this often leads to unexpected weird results. And because a drug needs to be refined and dose adjusted, doesn't make it trash. Actually, all drugs need to be refined and dose adjusted. And from what I've read, methylene blue has many useful properties which are often hard to come by. So I do have hope for this drug, and I do look forward to see how this will develop in the future. Having said this, please remember that I'm not trying to sell you a treatment, and I am not telling you that this is the ultimate answer to solve Alzheimer's. So, needless to say, please do not take it as medical advice. And most of all, please don't try this at home biohacking style. But I do hope that this video has shed light on an area of neuroscience which is worth understanding. At least I am glad to have come across this research. And here I will throw the ball in your court. If you feel that I have failed to mention anything in particular, or that my interpretation of a study is wrong, yeah, well, call me out. That's what the comment section is for. And if you found this unusual treatment for Alzheimer's intriguing, well, I would recommend you to watch this video next, where I go down the scientific rabbit hole of how frequency is being used to treat Alzheimer's. I tell you, I love this one. And with this, thanks for watching and see you in the next one. Ciao!